Right, let's ho ho ho. Hello and welcome to another episode of the Biofiles. This episode we're going to be doing another festive special. We have Did Symington, Jack Powell and myself, Ted Matthias. I've been discussing with my agent and they've asked for a rebrand, and so I'll go by Ted and or Ed. In this episode we're going to be taking you through some maybe tenuously festive stories to do with science, which we've been researching over the past couple of days to weeks. And so I think this week I'm going to start off, as I don't do it randomly, and I feel like I don't usually choose myself, and so this time I shall begin. We're actually going to begin on not a stop codon, but a start codon. Yeah, so for those of you who aren't familiar with the show, because we haven't recorded in over a year, we usually do stop codons, which are the end DNA sequence of a protein encoding DNA. So this time, though, we're going to do a start codon, because I'm going to quiz you before we begin the fact. Because what do you call those little green balls of vegetables which we have with our Christmas meal?
Sprouts.
Full name.
Brussels sprouts. Brussels sprouts.
And what are you saying, Dids?
Brussels sprouts.
So, are you saying Brussels sprouts or Brussels sprouts?
I'm pluralizing both.
I'll say singular.
Because
I'll say singular.
I say singular as well, but they are Brussels sprouts. There's an S, there's a Brussels, and then there's sprouts as well, and that's because it's homage to Brussels in Belgium, where they were a major exporter or producer of sprouts in the 13th century.
So
So
you're
Makes
telling me
sense.
that it's written wrong everywhere?
no, it's written correctly everywhere, and we're pronouncing it wrong everywhere.
Well, no, because if it's Brussels sprouts, it should be Brussels is sprouts, and then as in like it belongs to Brussels, it's Brussels is sprouts, and you would you put an apostrophe after the Brussels, right, instead of the extra S.
Yeah, true actually. Let's write to Big Sprout after this and see if we can get that corrected. See if we can get that corrected. I've decided to research a bit about Brussels sprouts because they're a festive treat, which are a bit controversial. And I, for one, am a fan. I think they are. I enjoy them with my Christmas meal. I just bought some more this
Me
week
too.
to do some research while in preparation for this podcast. Thought I'll have some with a few more meals. I had one with a Thai curry yesterday, which didn't quite fit, but but it was
What
nice.
did you do? What did you do with it? Just
I
boiled it regular.
boiled it with some rice, and then I mean Thai curries are so nice that you get the sauce from that as it is. So it did actually taste quite nice. Not as
Fair.
good as on a roast. But would you say that they taste bitter to you?
Maybe slight. Maybe that's if you're undercooking them. Maybe I feel they're still a little bit crunchy. I feel sometimes they're a bit bitter.
I don't know if Dids is thinking or looking into space.
Frozen.
Well, just like our last episode, the last time I had a Brussels sprouts was over a year ago.
Because a lot of the research, or not a lot of the well, there hasn't been much research on sprouts. There has nutritionally wise, but not so much on flavour profile. But apparently, 70% of people think that sprouts taste bitter, which I thought was bizarre because I don't really associate with sprouts being bitter. But you know, when you're younger and you either really hate sprouts or you like a bit more, I think it might be to do with that as well. And this can be found out to be genetically, so it's genetically associated whether you enjoy sprouts or not, because there is a gene called the TACE2 receptor member 38, which has a recessive allele, which then kind of means that you can't taste the bitter flavour if you get two of these recessive traits from both your mum and your dad. You inherit both the recessive ones, then you're going to be tasting or not tasting the sprouts bitter taste. But there's a bit of an interesting story going back into that, because apparently in 1931 there was a chemist called Arthur Fox who, it sounds a bit suspect, but he created a cloud of phenyl thiocarbamide (PTC) in his lab, and he noticed that one of his colleagues could really taste the bitterness of PTC, but he himself could not. And he thought, well, let's look into this a bit. So I think they assume that PTC isn't I hope that they knew that it wasn't kind of dangerous to be inhaling and tasting beforehand, especially they're making it as a cloud in their lab and then giving it to their family and friends. But they found out that there were some bitter tasters and some who weren't tasting bitterness. And then when they did some genetic analyses on this, they found it was due to this taste 2 receptor member 38 or TAS2R38, as it's called kind of genetically. And although PTC itself isn't in sprouts, there are kind of active agents within sprouts called glucosinolates, which contribute to their bitter taste. And there's kind of like a similar trend, because I think if you have that gene, then you can taste the glucosinolates in sprouts as you can taste PTC. But if you don't have it, or if you have the recessive allele, then you're not tasting the bitter taste. And so there's a bit of genetics going on there, which yeah, so next time you're having your sprouts, find out if you're tasting bitterness or not.
Have we discussed taste profiles before, as in with
As,
coriander?
I may have touched on it, but yeah, go shoot again.
I was going to say, I think last year for the Christmas episode, I covered something similar where there are a few papers that had discussed with your microbiome in your mouth can also affect the taste a little bit because there are some sulfur containing compounds, I think, in Brussels sprouts as well, and um depending on like the stuff you got growing in your mouth, basically, it can make them taste different depending on how it breaks them down.
It's a
Did
horrible
I
way to
don't know if that's
phrase it.
similar to what you were going to say. Yeah.
No,
It's
no.
kind of gross, but
I was just thinking. I've just there are several examples where genes are involved with flavor, or like a flavor preference. You're predisposed to like or dislike something based on the genes that you've got. And I just looked it up and confirmed coriander's one of them, where you're the specific genes that you've got. The gene involved here is apparently OR6A2, and some people hate coriander. They'll detect it in really small quantities in food, and they just can't eat it because it's grim.
it tastes like soap, doesn't it, for them? I think.
Yeah, yeah, it's really
mm.
weird, but it's very prominent. Yet you can use like the tiniest little amount. So that's really interesting. I'm just reading here about PTC, and apparently it affects kale, broccoli, and coffee. It makes them all extremely bitter as well, while others perceive these flavors as mild or pleasant.
Ah, I didn't know that it was in coffee as well, because the cabbage and kale and stuff, they're all part of the same brassica family, which I was then looking into because do you know about how brassicas like so that's kind of the family of plants where we get cabbage, sprouts, broccoli, kale, bok choy, like they've all come from the same family, but I think they've been bred to express different traits. So with sprouts, like that's expressing the budding or the buds and expressing them to be kind of quite flavorful and have a lot of energy in there. Whereas for broccoli, I think it's like the flowering part which they're making a bit more, and with kale and stuff, it'll be expressing the leaves more, so that you're getting more leafy content to then eat. And yeah, they just all come from the same kind of. I think mustard plants are there as well. They all come from the same kind of background of family, and they're all just interlinked. And yeah, so they all have kind of similar nutritional contents and PTC and so on and so forth.
I knew they were like roughly the same family, but I didn't realize they were so. It's almost like they're different stages of the same life cycle. Like each of them is focusing on a different part of the plant then
Yeah, so
for the brassica.
I
Yeah,
think that's the case. There's a
like Pokemon, like
just
the different evolutions.
just yeah, true actually. Which route you choose to take, which one do you choose to nurture? Yeah. One final thing on taste receptors, and then I've got another stop coding for you. But did we know that we have taste receptors in further places than our tongue? Um, so we've got the 5 taste receptors in our tongue, don't we? We've got the bitter, sweet, salty, umami, and is there a 5th one?
Sour?
Sour, yeah, and then sour as
Mm.
well. And so I don't know if the taste receptors in these other places are the same, but we have taste receptors in our airways, in our pancreas, in our gastrointestinal tract, which I can kind of understand that, in our liver, in our kidneys, in our brain, in our bladder, and in our testes for the male contingent. And they have non-tasting functions, but they are taste receptors, and I think they are somehow involved in some other like physiological functions. I didn't look too far into it because I don't think people really know why they're there, but yeah, we have them all over the place. And so that will lead us to the next stop codon. The first stop codon, as the first one's a start codon for this section. So I've already brushed over the fact that the Brussels sprouts came into Belgium started cultivating them quite highly in the 13th century. That's why they're Brussels rather than Brussels sprouts, which I appreciate sounded the exact same when I said it just there. Anyway, they're native to the Mediterranean region. I think they started getting big and kind of aware. There were maybe documents about them in the 5th century, which we can trace back. And so the first stop codon is, which nation in Europe eats the most Brussels sprouts each year?
This is probably like on QI where the buzzer would go off, but I'm gonna say Belgium.
Yeah, I was gonna say that as well, so I'll go the other way, and I reckon the Greek. I reckon the Greek love it.
Reckon Greek smash it in there. Yeah, you can imagine it with a bit of Greek olive oil and some.
Yeah, like a bit of flatbread, wrap it up with some hummus or falafel, whatever. Yeah, I reckon so.
Well, Jack, you did fall into the trap, and you're completely wrong. It's Britain. Us Brits. The UK is the largest consumer of sprouts worldwide. And apparently, in the UK, the sprout industry is worth £650 million, which is a lot of money for sprouts, especially as it's so controversial because a lot of people don't like them, and so that seems like quite a lot. And in terms of football fields, this is from the Royal Horticultural Society who are giving us these stats here. Do we know, or guess how many football fields worth of farming land is used for sprout growing
Where?
in the UK?
100
100 football
be my
fields.
guess.
Yeah, I mean, if it's that big business, 650 million, but then they're only small. I reckon 10.
10 football fields is producing £650 million worth of.
They work hard.
They are. Have you ever seen how sprouts grow? Because they are quite cumbersome. They're large. They're not really small.
Mini cabbage.
But they're growing on these like kind of big stalk bits, which need quite. Anyway, 3,240 football fields, which to me seemed like an outrageous amount of football fields. But I was then looking up how many football fields worth of well, how does this compare to the total crop production in the UK? And that's just 0.04% of the total crop space used of the UK. Using some government figures, converting football fields to hectares, and then seeing what proportion of that of the total hectares of crop land, and just 0.04%. So there's a lot of farmland in the UK.
I didn't think we had that much.
Yeah. Anyway,
10 was an underestimate, wasn't it?
Yeah.
Maybe we should edit that out.
Well, it'll make some listeners feel better for themselves for guessing 15 or something if you're guessing low at 10.
I expected them to grow like cabbages just in a line, and you'd have like 1,000 in a line, which would take up like an inch, and you've got many inches across a football field, right? So you could churn out a lot.
I think that's quite a naive way of looking at how sprouts grow.
Yeah.
I think you need to go back to your Royal Horticultural Society page and go back to your horticultural school and
I have some homework to do.
Yeah. I was going to say city living's put you out of the way, but you're not even city living yet. You're
We don't grow anything on the coast.
Yeah, true.
No vegetables survive on the coast.
Just marram grass and that's it.
Yeah.
One final thing about sprouts then is they are known to make people have some very smelly flatulence. And this is because they contain so these glucosinolates, which are what contribute to their bitterness flavour, they contain a functional group, so a chemical group on them, which has a sulfur containing moiety. So it's got sulfur in there. So this is bonded to carbon and some hydrogens as well. And this is broken down by the microbiota into the isothiocyanates. That's then broken down further into hydrogen sulfide, which is what the British used as a chemical weapon in World War One, as it's a very noxious gas which doesn't smell very nice at all. And that's one of the reasons why, sprouts can lead to some unpleasant effects down below. And they also have high levels of kind of cellulose and a sugar called raffinose, which we can't digest ourselves. We have to kind of leave that to our microbiota and our intestines. And they go crazy for it, and they produce methane and CO2 and hydrogen. But when it breaks this complex sugar down, it is then causing a lot of gas buildup, which why it might make some people more flatulent than others. And that concludes my my end on sprouts. So next up, we should be going on to DIDs for your session.
Great. So my session is, as usual, not quite as scientifically in depth as Ed's. I believe Ed devotes at least 30% of his PhD time to researching for the biofiles. Clearly, he's had a very busy year this year with his PhD, and has researched a lot into farting. Nice. I've taken the approach that this period of the year, December, January, is all about, reflection, regeneration, and growth. And because of that, I decided to focus on stem cells. I think we've spoken about stem cells before. I think.
They're a big topic, though. They deserve a lot of time.
They are, yeah. So I'm talking specifically here about rehabilitation in terms of literally regenerating muscles and regenerating joints and stuff like that. So the context here, do you guys know who Ross Edgley is?
No, I don't.
No. Ross Edgley is like an extreme sports athlete. He swam around Great Britain, did the Great British Swim 4 years ago now, I think, and where he swam literally around the circumference of Great Britain.
Wow.
Took him 150 something days. I think it's about 6 months to swim round, battling the currents like all the way up to Scotland, all the way back down. So pretty nails feet and really hard on the shoulders. Before then, he'd done like triathlons with logs on his back and all this stuff. So the point being is that he's just broken his body down. Like, this guy's a workhorse. He has a YouTube channel. You can hit him up. I'm sure he'll appreciate the thousands of biofile looking at his videos. Hopefully, maybe he'll mention us. But anyway, recently he's started to explore regeneration because his shoulder is a bit knackered. The things he's done is starting to take a bit of toll. I think he's 37, so he's getting on a bit. I just thought it was really interesting, so I thought we'd talk about it. Basically, the way it works, stem cells are, let's call them a miracle cell. They can generate an early stem cell, embryonic stem cell, or a umbilical cord stem cell is what's called totipotent. So they can generate any cell in the body. So muscle cell, brain cell, tissue cell, fat cell, nerve cell, anything. Cardiac cell, that's really important because cardiac cells can't regenerate. So to have these cells is super beneficial.
We start as stem cells. Is that correct? In the blastocyst, once the sperm's
Yeah, they're embryonic. Yeah. 8. There's a point where there's 8 cells, and those 8 cells are a blastocyst, I think, or blastocyte, and then they become everything in the body, effectively. Through multiple rounds of differentiation and division, they become every cell in the body. So you come from totipotent to, which is everything, and then you have pluripotent, which I believe is specifically body cells. So those totipotent cells will become the placenta and other things around, like the growth of the baby. And then we have pluripotent, which will become everything in the body. And then it differentiates a little bit further to like multipotent, and then you're into like, this is a muscle stem cell, which can go and produce any muscle cell, cardiac or thigh or something like that. And then obviously you've got nerve cells, bone marrow cells, everything else like that. And at that stage, they're specific. So you can't take a muscle stem cell and make a nerve cell from it, unless you were to reprogram it. But biologically, you couldn't do that naturally. So what's really great about this stem cells of regenerative therapy is that when you get to the ages of 40, 50, 60, like you get a little bit later on, your body's breaking down, you recover slower and not as effectively as you would when you're younger. And a big reason for that is that you have a reduced number of circulating stem cells or a reduced ability in the body to generate stem cells, because we've only got a certain like amount that
Okay.
we can do. It reduces as we get older.
So
So
is
to
that
inject,
a finite amount? Sorry, or do we? So once it's like, do we have X number of stem cells in our life? Kind of like how you have X number of eggs if you're a female in your life,
yeah, I
or
don't.
do they regenerate kind of more? Do they get produced more slowly the older you get? So
I
new
think it's the
stem cells
latter.
get produced more slowly.
I think it's the latter. Don't quote me on it, but I don't believe it's the same as the egg situation. Because the
Okay.
egg situation, they start with a million, and then eventually you'll run out.
Yeah.
If you can live that long. But yeah, no, I don't think it's like that. I think it's
Just
the.
a nice
I think.
morbid twist on the already quite morbid topic.
Yeah. Yeah. No, I think your ability to produce them decreases. So
Okay. Yeah.
naturally in the body, what happens is when you get like an injury, let's say like arthritis is a really good example. This is where arthritis is where most of these regenerative stem cell therapies are like coming in. You've absolutely shattered your joints; they just can't take it anymore, and you've got like a lot of pain operating your hands, maybe your elbows, knees, lower back, stuff like that. What happens is your body starts to release a chemokine or a cytokine called SDF1, and SDF1 stands for
Oh, this is the suspense.
Stem differentiation factor. I definitely
stromal.
get bonus points if I guess that. Stromal.
Stromal. This is the first step. This is the first stop code on, guys. Let's jump in with jingle. What define to me SDF1? Does anybody know what
Is
SDF1
it
stands for?
stromal differentiation factor 1 by any
Ed,
chance?
what's your attempt?
Oh, I probably would have guessed Santa doesn't find one, and
You
that's it.
done?
Yep.
Ed, Jack, you were the closest, but Ed, you were by far the most festive. It's stromal cell derived factor 1. And stromal cell derived factor 1, SDF1, is a chemokine protein which is excreted from damaged tissue
Just to
into
say,
the
just
bloodstream.
to quick injection. So chemokines are like messengers, aren't they? They
Yeah.
leave cells as messages to other cells.
And they want to call about a response. So in this case, we want to start repair. We want to initiate repair.
Okay.
So this SDF protein floats around the blood, makes its way to the bone marrow in an adult human, where our stem cells reside. Repair stem cells reside in the bone marrow. This stimulates the release of stem cells, which in itself is its own little process. The stem cell will differentiate into the appropriate stem cell type. So if it's a muscle that's damaged, it'll become a muscle. If it's a joint specific damage, then it'll differentiate into a stem cell that is joint specific.
Would you have liver
And then
specific ones as well? Like we know that liver regenerates a lot. Is that?
Yeah, liver stem cells. I don't know if you will do it on it. I don't know if you can make your body generate a nerve cell on its own, but that can be done. Cardiac is written out; they don't regenerate. Brain cells, nerve cells. I don't know. I don't know. Off the top of my head, don't know. Feel free to jump in. I am talking about a lot. I'm almost at the end of this process, but effectively, the SDF will initiate the release of stem cells from the bone marrow, and then the stem cells will migrate throughout the blood, and then they basically do. Do you remember chemotaxis in bacterial migration? Do you remember chemotaxis?
Oh yeah. That's with antibodies and such, isn't it? They're involved in chemotaxis.
Yeah. Effectively, antibodies will follow like a concentration gradient of chemicals towards that's how they detect where the baddies are and they go get it. In that sense, the stem cells do the same thing. They locate where the SDF is being excreted and then they go into the place to go and basically bring about the repair. So they arrive at the site, they differentiate, they divide, and then they repopulate that site and repair the area. and then eventually, you're good again.
So how does this relate to Mr. Man who went across the not just a channel, went around the UK? So is he using these cells, or is he looking into them? Are there trials in these cells, and is he one of the participants?
So, as a regenerative medicine, they're actually used like internationally now, as of like a few years. I think they've been used for a few, maybe a decade now. But they're definitely in use now in 2024. Mexico, South America, a lot of places there. They've got a lot of stem cell clinics in there. And But he documents it in his YouTube video. He went to Utah in the US, and effectively the reason for me describing the process of release and differentiation and stuff like this is that because we understand science, you can now extract and culture stem cells and then inject them at the sites that you want them to work. So, for example, if you've got knackered shoulders, you can get them injected into your shoulders, and you'll have a localized like repair in the shoulder, and it will do it. So if your left shoulder hurts, you go get your left shoulder fixed. Your
Does
right
this
shoulder hurts, go get your right shoulder fixed.
have these been through trials and stuff, or is it just
Yeah,
private
yeah.
people?
Yeah,
Wow.
no, these have been through trials. There's academic publications out there at the moment. I don't believe it would ever be something you do on the NHS because it's so expensive.
Okay.
But the science is there, the medical science is there, and it's a really big thing in like people who are really conscious about aging and trying to perform physically at like those later ages, like 40, 50, 60. I know they're not necessarily old, but they are a bit old to be doing like climbing Mount Everest or to go and swim around Great Britain again, maybe. Um, so
Wow.
yeah, I
Yeah.
open the floor to discussion.
So what diseases can these be used for? Do you know? So if you're using them instead, just like not just to improve your physical abilities, but say you have kind of let's say like a neurodegenerative disease where like kind of Alzheimer's and stuff, you're breaking down your healthy cells within your brain. Could it be explored for that? Do you know?
Potentially, the theory's definitely there. I do know of a few papers that have cultured stem cells to like neurites and to regenerate neuronal pathways and stuff like that. But obviously, when you start to do things in the brain to get that as a treatment that you can go and get, it's going to take years. I think. I don't know if it's there currently,
Yeah.
but I would imagine it would take years. The biggest application for me, that is like the clearest, is as I was saying during that description that cardiomyocytes or cardiac cells, the cells of the heart, they very famously don't repair. So if you have a heart attack and you develop scar tissue in your heart, that will not go away. You effectively have a percentage reduced output from the heart forever because you can't repair it. The application of stem cells, of exogenously produced or at least exogenously sourced stem cells, so they're coming from outside the body, they're given deliberately by somebody. We can get into how you source the stem cells, but what I'm saying is that they're administered. You could, in theory, inject stem cells into the heart of a heart attack patient once they're stabilized, etc. And you could see regeneration of the cardiac muscle back to healthy or even like enhanced cardiac function. It's the technology effectively means that you can regenerate anything you need to regenerate, if you've got enough money.
Got keep. I think keep your eyes peeled over the next decade or so to see if that can get into clinics and more conventional medicines as well as just I say conventional, more mainstream medicines as well. And maybe Father Christmas already knows about this, as he probably has to put in a big shift on the 24th, the night of the 24th, morning of the 25th each day. I'm sure that's kind of going to bugger his bones up quite a lot. And so maybe he's injecting stem cells into himself and the deers to try and help them along with their recovery. And
Sensitive juicing.
and I think with that, let's move on to Jack.
Yeah, so I've got a topic that's not so much Christmassy, but it's definitely a wintry one. So I was looking at a paper on how frogs and fish and stuff like that manage to hibernate over the winter. So obviously you have frogs. They're living in a very wet environment. They're made up of water, much like we are. But then they somehow manage to survive the winters when it's sub-zero temperatures, which you would think they would just turn into a green popsicle.
Yeah.
Um, but they have some quite cool evolutionary adaptations that allow them to survive over the winter. Um, so I don't know if you guys have worked much with freezing down cell stocks before, but what we tend to use is traditional cryopreservants like DMSO, which is dimethyl sulfoxide, or you can also use sugars like glycogen. And basically, what these do is they intermix with the water, and they raise the freezing temperature of the water because the water is now I'm not doing a very good job of explaining this.
Diluted,
But
is
yeah,
it?
yeah. So essentially, what you're doing is you're putting these molecules in there, like your DMSO or your glycogen. And what this does is when the water starts to cool down, it finds it harder to freeze into that crystal structure that ice has.
Oh.
And so what this does is it means you can go down to a lower temperature without getting those ice crystal formations, which would be what would kill the frog if it didn't have these in.
Because these crystals
Um,
begin to lyse the cells, is that right? So they were like kind of stab holes into the cells, which are intermixed within this water. They'll then break open, and obviously, if you're breaking open your cells, and you're gonna have some problems. So these cryopreservatives prevent
yes.
those for those cells getting broken.
Yes, so that's what we use typically, but they do have their downsides because you have to use fairly high concentrations of them to get this effect, and they're just sort of unwanted chemicals that your cells, generally speaking, don't want mixing in with all the other chemicals that are racing around in there. But what the frogs have, and some fish and insects have this too as well, I think, is they have specific proteins to counteract the freezing rather than uh, these traditional cryoprotectants, which are smaller molecules. They're sort of just regular chemicals that you could find floating about in the environment normally, whereas these are specific proteins which they've evolved over millions of years, millions of winters,
Oh,
to
cool. Yeah.
help them survive. And they so a benefit of that is they avoid a lot of the toxicity issues that you would get with those other cryopreservants, because although frogs are very different to us, they do have a fair amount of similarities in their cells between human cells and frog cells. So they,
So how do the proteins work?
yeah,
How do they prevent the water from freezing as it usually would? And would they lower the freezing temperature of water? So instead of freezing at 0, would it freeze at like -10?
yes, yeah, exactly. So rather than sort of crowding out the water and stopping the formation, they're still trying to work out the exact mode of action of these proteins, but they know it's just something to do with their function that allows them to stop that ice formation, um, but at much lower concentrations. So yeah, you could go down to -10 degrees. These cells wouldn't freeze up because they've got these protective proteins. Um, but yeah, that was what this paper that I found is focusing on. They're trying to work out the structure of these proteins so they could try and replicate, make some synthetic ones, and then this could potentially be used for transplants, for example. So if you are getting a heart transplant or liver transplant or something like that, we currently have certain methods of slowing down the metabolic processes in them so that we can get them to the recipient in time without them just conking out. And with these cryopreservants, we could potentially do longer distances of storing and transporting these organs by actually freezing them in this non-toxic cryopreservant mixture.
And do you reckon
No,
that'd make
that's
us? Do
quality.
you reckon that'd make us frog-like as well? Do you reckon we could be a bit more,
Ribbit.
ribbity?
Wanted to say, do you know if these proteins are always present in the blood or the cells of? Well, firstly, are these blood-borne proteins or are these cellular proteins?
So these are cellular proteins, so they're permeating all of the cells. in that frog's body, so that you don't get any ice formation within the cells, because as Ed said, that would just rip them open and burst them,
Yeah.
and they would wake up not feeling very well in the spring.
Yeah.
Um,
So
so I
you,
think there is some uptake in uptick in their production as winter's coming on, but I think they are always there at some level in the cells.
okay. That was going to be my second question. Is that are they excreted like seasonally, or are they constantly present?
I think it's a mix. Like I said, the amphibians are they've been around for billions of years. They're a very old order of animals. So they're actually a very diverse set of proteins. These it's not just, five or six different proteins that they've developed over the years. It's a huge amount, and across multiple species as well, not just amphibians. You've got fish, insects. So I think some of them are expressed in different ways. Some of them are there all the time, and some of them are just produced when it starts getting a bit chilly, I think.
And do frogs still like jump around in winter? This might be quite a naive thing to say. Do they or do they just kind of go into like a form of hibernation as well, like a stasis? Do they just find somewhere quiet and chill?
They're cold blooded, aren't they?
Yeah. So
Yes, yeah.
they'll need the sun to start warming up. But
Yeah, I think what they do is they'll find a pond or something like that, and they just stay underwater and go into a sort of hibernation mode. Um, Obviously, the pond stays slightly warmer than the outside temperature because you have the ice that forms over the top, which insulates the pond. Quite
Mhm.
often, it will stay just above zero throughout the year. But I think even if you froze one solid, I think because these cryoprotectants, as long as it's a slow thawing, uh, whenever the springtime comes, I think they can actually survive that as well, which is pretty cool.
In
yeah.
terms of in terms of metabolic demand, do you know anything about how metabolically demanding it is to make, secrete, and like populate the cells with these proteins?
Yeah, I haven't read anything on this in particular, but I don't think they're particularly large or complex proteins. So I don't think there is any huge
Yeah.
metabolic requirement, and they'll probably produce these proteins during the summer or like during the autumn when they're still metabolically active, and then once they go into hibernation, their metabolism starts slowing down. They're kind of sorted and they're safe.
Yeah. No, I just asked because that's so interesting that like bears hibernate throughout the summer, and Ed, you mentioned hibernation with this frog, and I'm just wondering if like that's the right word because obviously when bears or mammals, I believe they are mammals, will go into hibernation, they use uncouplers, don't they, to keep their body warm during like the winter. So instead of secreting cryopreservants or something that would stop their blood and cells from freezing, they just keep their bodies warm um, by effectively uncoupling metabolic processes to generate heat as opposed to energy for movement and other stuff. So it's a real good example almost of divergent evolution where we have the same goal and we've done it in a completely different way. I'm gonna fill my blood with antifreeze, and you're gonna turn on the central heating. Like it's wicked.
That's cool, isn't it? Yeah.
Excuse me, gentlemen.
And that's a Christmas. It's a Christmas miracle. That's a good point though, did saying that the instead of turning on the central heating, they've gone for the cryopreservatives. And I assume frogs are older than bears, so wonder why bears didn't opt for the cryopreservation themselves.
Well, they're warm-blooded, warm and cold-blood,
Hmm.
right?
Yeah.
So warm-blooded, they can generate their own heat. Cold-blooded, I don't believe can generate their own heat. So that's why they're cold-blooded, right?
It's kind of cool blooded, yeah.
Yeah. So yeah, they couldn't.
Hmm.
They've had the same problem, and through diversity, we've achieved a plethora of options to solve it.
Quite right. And Jack, you say that fish and other insects and stuff have them. Do they? You're saying it's a complex. They're quite complex in the family of proteins. Do they share the same ones, or do different animals have different sets of these cryopreservatives?
I'm not too sure. That's a bit of a gap in my research. I think some of them probably would have come from the sort of fish lineage. They would be even older than frogs. And they may have evolved in fish and then carried through as they evolved into amphibians. Um, but I think, given the fact you've got so many species with these proteins, there's probably a lot that started on their own and they're completely unrelated.
So we probably have quite a
Um,
large market to tap into to see to improve these, as you say, kidney, well not sorry, not kidneys, but organ donations and improving,
yeah,
yeah, organ time.
yeah, because I think some of them do still have slight toxicity issues when you put them into human cells, but we have so many to pick from. It's easy to sort of work your way through them, find some that aren't, or you know, through structural research of these proteins, try and work out which ones aren't toxic and try and make some new ones ourselves. Um, and who knows, maybe even start freezing ourselves if we want to time travel to the year 3000 or something. Instead of dying of cancer at 50, you could just freeze yourself and wake up 1,000 years time, like
But
Futurama.
that's what I
I don't
mean with
know.
like the the metabolic demand for cryopreservation is so much lower. It leads itself to longer term storage almost.
Yeah. Oh yeah, like if we wanted to do a similar thing to bears for like long term store, because there are a lot of people up doing that, aren't they? Where they're freezing themselves and they're hoping they're going to wake up in 100 years' time when they've cured cancer or something.
Because
I
Yeah.
guess this could potentially help them with that.
their blood is getting flushed out for cryopreservation, but probably more like
Yes,
commercial things which you'd use in your car and stuff to prevent
I
freezing
think it is.
rather than some
Yeah,
nice healthy proteins which seem a bit more palatable
Mm.
than, I don't know, just some straight chemicals.
Well,
I think yeah.
that would be a great way of cryogenically preserving someone, because obviously you can't just drop someone in liquid nitrogen, because you would have that cold, the water would freeze, you would have necrosis purely from like fractured cells because of the ice crystals, right? You would lose a lot of tissue. I mean, you get frostbite, you'd be really severely impacted. So if we did want to pause people for ages, then research in that area would be sensational. Does anybody know how people are being frozen? Because people are being frozen. They're having tissues
Mm.
frozen and preserved for the future. Do you know how that's done?
It is remarkably sort of blunt force. Um, yeah, you would think they've got it down to some highly sophisticated science by now, but it sounds like they are literally replacing, as you said, replacing their blood with DMSO, which is a pretty toxic chemical,
That's really toxic.
um,
When
and
we're
then just dunking them in liquid nitrogen and hoping that they'll wake up on the other side.
Well, and
Um,
they'll deal with the frostbite on the other end when medicine's
yeah,
advanced.
Yeah, they hope they found the way to bring them back by that point. So Jack, this has all been very scientific. Is there anything festive to bring us back to the this festive time of year?
yes. So I do have a stock code on. It's not the most trustworthy paper, perhaps, but it is very festive, very light-hearted. So I thought I'd share it. I think it's from a lab in Denmark, if I remember correctly. But they wanted to identify the Christmas spirit, and they have a functional MRI in their lab, which, for those who don't know, is a way of tracking the glucose use within your brain. So the idea being that if they show you images or something, depending on which areas of your brain that those images activate, you'll be able to see that with this functional MRI. You'll be able to see which areas of the brain are being activated.
Because they're
And
using the
so what they
glucose.
because they're using the glucose, yeah, you'll be able to see that sudden increase in uptake of glucose in certain areas of the brain. And so what they did is they got a group of people who celebrate Christmas and a control group who historically haven't celebrated Christmas. And they showed both groups images of sort of Christmas trees, a turkey, uh, a nice little village with some Christmas lights. And they measured the differences in brain activation between the two different groups. and they did find a fairly consistent, actually, network of areas of the brain which are activated by the Christmassy images in the Christmas group, but wasn't found in the non-Christmas group. So they are, yes, they're claiming that they have indeed found the Christmas spirit.
Oh wow.
Yeah, I
So the
should have made that a question, shouldn't I? Instead of just wait. So stop and
gap.
listen.
Yeah.
Stop and listen.
Very festive. Um, oh wow. So is that saying that there's a certain area of the brain which lights up, so it's using energy when people who celebrate Christmas look at Christmas-related images, but those people who don't celebrate Christmas, that area of the brain does not light up.
Yes, yeah. So it seems to be related to the sort of memories they have of previous Christmases and the positive feelings and memories associated with those kinds of images. It seems to be lighting up those areas, whereas people who historically haven't celebrated it don't get that kind of response.
All right. Should we start any more before we wrap up? Reaching a nice natural there. And so, okay, that leaves us to say thank you very much for listening to this festive episode of the Biofiles with Did Symington, Jack Powell, and myself, Ted Mathias. We hope you've enjoyed listening, and we're going to try and make these a bit more consistent in the new 2025 year, although there's no promises. So we hope you have a lovely festive break if you're celebrating Christmas or not, and you get some time to relax before the new year begins. And we shall see you in the new year. Thank you very much.
Merry Christmas.
Happy holidays.